Hi, my name is Dylan Terstege. I'm a PhD candidate in the EP Lab within the University of Calgary's Hotchkiss Brain Institute. Today I'm going to discuss my research into learning and memory and FASTMAP, a tool which I've developed to further this study. Memories are stored as networks of brain activity. These networks are often dispersed throughout the brain across many neuroanatomical regions. It's important to note that these networks are not stable. They're dynamic. Over time, memory storage reorganizes. This shifts the relative involvement of certain neuroanatomical regions in the maintenance of memory networks. During my PhD, I've been studying how the generation of new neurons contributes to this reorganization. In order to look at memory networks, I needed a proxy of brain activity during memory recall and a means to map this activity across the brain. The first issue is fairly easy to address, as there are several well-established histological markers of cellular activation, which can serve as my activity proxy. The true test comes in terms of the mapping of these markers. While commercial options exist, they are often inflexible to tissue type, or inflexible to regional organization, or require extensive computational backgrounds, or they're just plain expensive. With all of this in mind, I decided to develop my own open source tool to map cellular labels of interest to an anatomical atlas. FASTMAP, which stands for Flexible Atlas Segmentation Tool for Multi-Area Processing, is an open source plugin which I've developed for the free-to-use platform ImageJ. FASTMAP guides users through the generation of custom atlases which they can base on industry standards irrespective of tissue type or regional organization. Without the need for extensive computational knowledge, FASTMAP facilitates the registration of biological image sets to these custom atlases and segments the markers of interest within each region to report regional densities. On the right side of the slide, I've included some screenshots to illustrate the registration process while using FASTMAP. Users first identify their atlas of interest. In this case, it's a fairly detailed sagittal atlas of an adult mouse brain. Next, the user defines the folder containing the image that they would like to register. One of these folders should contain the label of interest, while the other contains an imaging channel in which anatomical structures are clearly visible for easier registration. The user is also presented with the option to analyze their data as object counts, meaning the number of cells per region, or as volumetric analysis, in which the total area of the label of interest is reported per region. In both cases, this output data is presented with the total area of the region itself, allowing for the calculation of regional densities. When running the analysis, a target image populates the screen, and users are asked to identify which atlas plate most closely resembles the target. The custom atlas plate will then be applied to the image, and one by one, regions will resize themselves linearly to better align with the target. Users can then refine the registration manually in a free-form manner to ensure a perfect registration. Once satisfied with the alignment, the registration is then applied to the imaging channel containing the labels of interest. I'm hoping that this tool will be beneficial to the open science community in several ways. Firstly, I will be uploading the tool itself along with several resources and guides for users to publicly accessible repositories, such as GitHub and the CONP portal within the open science framework. Secondly, I believe that FASTMAP can foster transparency and reproducibility in the image registration communities by facilitating the sharing of custom atlases. When publishing a project, FASTMAP users can easily upload their custom atlas plates to further illustrate the regions of interest that they're actually registering to. In closing, I would like to take a moment to acknowledge and thank my co-authors, particularly Daniela Obo, who helped with the development of FASTMAP, and my supervisor, Dr. Jonathan Epp. I'd also like to thank the Cummings School of Medicine's Advanced Microscopy platform for assistance with image collection. Finally, I'd like to thank the Canadian Open Neuroscience platform for the support which I have received during this project. Thank you all again.